Hello everyone, I am Kang Min Lee and I will introduce you the paper Temporal Dynamics from a spatial snapshot. So let's start with the abstract. Are you done? Yeah. How do you think about this abstract? No cap sentence. No cap sentence, really? Do you think it is easy? No. No. However, yeah. So that is our gap sentence. And so to follow their population over time within the native context argument sure lacking because the biopsy occurs on the single snapshot. So throughout this general club, the single snapshot is the keyword of today's study. So okay, let's have no question or comments. Let's start. So let's try with some simple illustration. So there are two people and both enjoy drinking and smoking. So they visit hospital, and in hospital they will do kind of biopsy like 조직검사. And through that, the first man had this result. So this one is normal tissue, and the red one is the tumor tissue. So looking at this result, you will say, they're still okay. So the kid in street drinking and smoking, and they will face some bad consequence. And there is one more guy. He also had the same result, but he was kind of suspicious. So he tried to predict his future, which is unseen in this form. And so because he predicted this future is very bad, so he quit this drinking and smoking. So there is a quiz. What is his job? He's prophet, or he's GPT professional, or he is mathematical biologist. What will be the answer?
Number
Second.
two.
Okay. So the answer is mathematical biology. So he used the snapshot data to predict. But how he did it? It was not the fan of the contact lecture. This is our most read book. So from this data, what he did was only he built some dynamical model, and he applied the dynamic, which is called the phase portrait, and he also simulated some stochastic system. So this is really the summary of today's journal club, and today I will go step by step. Let's start with how they build this model. So when you have this snapshot data or like biopsy data, you want first to know the mechanism. You want to like select some mechanism for the cell dynamics. So you can think of division, and also you can think of cell death, and maybe you can think of cell migration and trans differentiation. But for this specific paper, they don't really consider these two mechanisms because they are very slow relative to division and death. So with these two mechanisms, they build an equation on very naturally, they write down the inequality and some equation each corresponding to division and cell death. So what we usually do with this model is we will infer the parameter, the alpha and beta. And this should be learned from the data. And very usual thing we do is that we collect temporal dynamics because we want to know the dynamics of the data. So something like this, we have each data point here. And then we fit the data to know the alpha and beta. However, our situation is that we only have this one snapshot. Then how do we know? So instead of this full dynamic, what we use or they use is they use the instantaneous dynamics or instantaneous rate of this API. So you may know which cell is growing or which cell is going to die. Let's try. I will explain this with very specific example. Now, okay, this is your tissue sample, and you will focus on this big red one first. And first, you should decide the state of this one specific cell. But for some reason, they only decided for binary state. So they will say if division is going on or not. And they will say that this is going on or not. This is all binary. And one more thing to do is that first they set some current neighborhood of this specific cell. And they count the composition. They have 5 rectangular cells and they have 2 triangular cells. And almost done. You only need to repeat this process for every other cells. This cell and that cell. Repeat and repeat. After you end up with this kind of summary of the snapshot. So here you can see for each cell, you will know determine the cell type and determine if they are on division or on death. And you will count the number of the other cells in the neighborhood. Any questions?
Okay.
Yeah.
I have a simple question. How do they decide the radius?
The subtle point, they had some standard, but they also tried the other different radius, and they say robust. But I think there is some typical length scale in because I will explain later very briefly, but so there should be cell-to-cell communication between cells, and that cell-to-cell communication will determine their growth and their death. So if they are too far, there is no possibility that communication can be done. So there is some appropriate scale, length scale.
In the data set,
Yeah.
they can analyze the cell type, division, and density.
Actually, we only do this. And I will explain later. But for their death rate, they just fix one death rate as mean of division rate, but they determine each division state.
So they can do that.
Yeah, they can do it.
Yes. Spatial trans.
Spatial
protein.
protein.
They have some protein.
Yeah, protein that only appear during their division stage.
I see.
Actually, I really omitted many parts of the biological details, but please ask if you are interested.
A very naive question, but is there any possibility that there is no both division in that?
No post division death. I haven't think of it, but
But not that important.
maybe. Okay, if there are no more questions. And then from this snapshot summary, they build some statistical model. It is very simple. And you can see this probability. The first one is the cell type, and the second one is their state. So this probability means this red cell in the state of division is ongoing. So they want to write this as function of their environment. As I mentioned just before, because the cell-to-cell communication and like their environment really affect their division and growth, division and death. They want to write it in function of the environment. But really simply, they just consider this environment as the composition of neighborhood. So this function F can be any model, but in this paper, we use the logistic regression for the simplest choice to calculate this probability. So let's consider at T0, we have some observation, that is the snapshot. And in this framework, we can determine the probabilities of each cell's division and death. For this, division is 0.5 and death 0.2. And you can also do this for other cells. In fact, every cells. And for the next time step, which cannot be observed because we have no observation after that, you can just sample according to these probabilities. To simulate this state transition after time. And this time you can also calculate the probability of gain sample, and simulate the system. We will at last have the simulated trajectory, something you may see on like Gillespie stochastic simulation, but be careful, this is not really Gillespie algorithm. It's just more simple version of the simple template.
The function they use only number of green and red. It doesn't care about the division.
In this here?
Yeah.
No, the division should be they want to predict the division
Yeah.
as probability. And here they only use the cell count
Cell count.
as predictor.
It doesn't matter whether their neighbor is in division.
Their neighborhood in division or not. Yes, they didn't consider that.
셀 타임만 고려. 네이버.
그것도 중요하긴 한데.
And also, does the neighborhood include itself?
No.
It doesn't.
Yeah.
So then that means it's someone's fate is not affected by their own state.
Yes, but because the state information is not accounted in this model, let's see, only the composition. In this situation, yeah, that is considered.
Here, time is the real time is just some sampling step.
So yeah, it's just sampling step. And in fact, when they want to know real time scale, they set this time scale as so there is there's some biomarker which increased during the period of division. So they use the time and typical time scale for estimation.
So they set the
Um,
one step as a DT.
Yes.
Uh,
Okay. Now, the next thing they can do is then put these cells into mixing machine, and they create some cell smoothie. So this cell smoothie is composed of 0.75% of green cells and 25% of the red cells. This is well mixed condition. When we come to the well mixed condition, we can use ODE for describing this system. So you can write down the ODE model for the red cell, which is the rate is replaced with the probability we just computed in the previous slide. And also the probability of bud deaths. So again, is in fact function of the composition, green or red and green or red. But in this case, because they are well mixed condition, you can just put these values here. And also for the green cell, you can repeat the same procedure and this. And finally, you can do density of green and density of red. So let's think one point here. According to this equation, you will make some vector field. The x-axis will be the rate of green. The y-axis will be the rate of the red. And you can repeat this for the other point, and we will draw some line. So this is the thing we learn as the base portrait of this system. So really good thing about this base portrait is that it is kind of a summary of the dynamics of this system. And if you want to know if some system is here, then this will go, and you can follow this arrow where how the system will evolve. So as a small summary, this can be done by the snapshot summary. They just do some statistical model and put them into the OD model to make this phase portrait. And actually, this is the really old method of their paper, and I will explain you some application of this method.
How did they calculate the composition? So did they assume that the well mixed condition is satisfied within some neighborhood, or is it?
If they consider this kind of situation, they just get it all together, not considering some specific neighborhoods.
그래서 잘못해서 스냅샷을 2개 해서 미래를 예측하는 거죠. 본 상태만 됐지.
하나 해서 이 정도만.
그러니까 테이블 하나에 저 함수 하나 피팅했어. 그리고 저 함수 가지고 이제 전체 웰믹스를 가져오겠어요.
맞아요.
잘 섞였을 거라고 하는 건데 스페이셜 정보가 있는데
그러니까
저렇게 또 다시 한번 얘기해야 되나?
그래서 이 스페이셜 정보는 딱 앞에 있는 시뮬레이션 이런 걸 만들 때만 써요. 이런 알고 있지 민겨버린 거죠.
아니면 약간 네이버후드 레디어스 하나 정해가지고 그 컴포지션들의 그 평균을 좀 쓰거나 매력 공식하면 안 돼. 진짜 다 이렇게 컴포지션 쭉 압축하지 말고 레디어스 컴포지션 구해서
약간 되게 컴파트먼트 모델.
평균을 구하거나 컴파트먼트를
그 사이
이렇게 가봐야 되는지 모르겠네.
이게 다양한 PDF가 써지잖아. 스크래프가 위치마다 나와 있어.
그러니까요.
위치마다 함수가
근데 PD를 써도
베이스 플레인을 그려야 돼서.
사실 막 얘는 확실히 페이지 플랜을 그리려고 하는 것 같고. Okay. There's no more questions. So this is summary of their method, and they call this one-shot tissue dynamics reconstruction. One-shot because they only use the one snapshot. So they apply their method to breast cancer data. So in this data, they consider three components: fibroblast and macrophage and cancer cell. So they took the snapshot of the tissue, and as their method, they plotted this phase diagram. So this line means their null line dynamics. So null line is just that their rate becomes zero. So in this line, the rate of fibroblast change is zero. Okay, and they compare this phase diagram with real in vitro mouse tissue data. And in this data is 4-day observation. So each arrow corresponds to 4-day observation of one in vitro tissue. So this is really difficult to get data, really expensive data, and this is like relatively cheap data. So they noted that there's 2 points we need to focus. First, in this experiment, this is a previous study, they find out 2 like stable points, which is hot fibrosis and cold fibrosis. So this means they have high fibroblast density here for the each point are attracted to these 2 points. And also in their phase diagram, they portray the hot fibrosis and cold fibrosis are reproduced in their just from their snapshot data. So they can uncover the blind dynamics without the long-term observation. And the hot fibrosis is hypothesized to be clinically important in the cancer development. So they consider if they counted 1% the density of each cell types. And then they ask whether this density is closer to hot fibrosis or cold fibrosis of their cell, of their tissue. So they just divided people into 2 groups and they compare their survival probability according to these 2 groups. And it really turns out that higher mortality observed for hot fibrosis group. So this indicates that if we can know someone's position on their face portrait, we can like predict or explain their mortality rate. Let's go to the next example. In the same breast cancer data, they have also the snapshot for the T cell and B cell. They also draw this phase diagram, and this phase diagram is really interesting. First, they have the stable fixed point at 0. And one thing to notice is that after the T cell cross the threshold, then suddenly they go to big turnaround here. Or T cell increase and then T cell decrease and go to 0. They simulated these dynamics, and you can also see that T cell first increase and B cell decrease. And this kind of phenomena is really often termed as the excitability. So excitability, so when you cross some threshold, the system suddenly excites to make this kind of pulse or spike. So they found out this system is some kind of excitable system. And usually this kind of excitation, or I will just term spike, can happen when the T cell cross the threshold, such as adding T cell. So if you add the T cell, the system will make a spike. And if you again add the T cell, again does another spike. But if you add T cell, but if the time interval is too short, then the system cannot make another spike. This phenomena is called some kind of refractory period. And it means that some time is needed to make another spike. They also tested this phenomena in their model, and you can see here, adding T cell immediately cannot make another spike. So this again demonstrates that this OSDR also have reproduced the refractory period. And this is the binary example. They also brought this breast cancer data, and but for this time, the treatment is done on the patient. And after this early treatment, they do biopsy. And then after the whole treatment, they can divide people by response group and non-response group. So that means in this group, people, the treatment response was good and treatment response was bad. And for both group, they apply OSDR respectively. So they can build two models, model 1, model 2. And they did stochastic simulation with various initial conditions for each model, the response group and non-response group. It turns out that, okay, this T-cell trajectory was quite similar. However, but the tumor cell trajectory was really higher in the non-response. But in response group, it decreased at the end. So this demonstrates that the effect of treatment can be inferred from the snapshot right after this early treatment. So this is our last summary. OSDR can infer. This is the method that can infer dynamics from tissue snapshot. And it converts snapshot summary into dynamic model. And they made base portrait or stochastic simulation to reveal the characteristics of cell dynamics within tissue. So, thank you for listening. That's all.